All right, so this is our video on translation specifically. If you recall, the previous video you watched should have been describing transcription. And so remember, if you recall, transcription is making an mRNA copy from a DNA template, specifically from the template strand of DNA. And during translation, we also briefly introduced different kinds of RNA. So during transcription, you're making an mRNA copy, which stands for messenger RNA. This RNA actually includes the instructions for a specific gene, and those instructions are for what proteins that gene should be making. rRNA stands for ribosomal RNA, and this is important because ribosomes are the site of translation. So without some ribosomal RNA, you would not have those ribosomes. And tRNA stands for transfer RNA. So this RNA is responsible for bringing or transferring those different amino acids, right? If you recall from earlier lectures, like we're talking about beginning of the semester lectures, when we talked about the subunits of proteins, which remember are things like amino acids, right? So many amino acids together make up a polypeptide chain, which then makes up certain proteins, right? And so that transfer RNA is bringing those amino acids to your ribosome, which is the site of translation. Those ribosomes are going to help bind those different amino acids together to create certain proteins. Okay, so ribosomal RNA, it's our actual most abundant type of RNA in your cells. Our cells have lots of ribosomes, and ribosomal RNA is found pretty much everywhere. So a bacterial cell wall is going to have typically several thousand ribosomes. A human liver will contain several million. So we have ribosomes everywhere. We have lots of ribosomal RNA. So ribosomes themselves, in addition to being a cellular organelle, which you guys should already know, they're composed of rRNA and proteins, and they're organized into these two subunits. So you have a large subunit and you have a small subunit. Pretty easy so far. Remember, ribosomes are the site of translation, so this is where the action happens. We have transfer RNAs or tRNAs, and they're going to actually shuttle your amino acids to the ribosome during translation. So your amino acids will attach up here. It could be all kinds of amino acids, lysine, methionine. There's so many. They're on your chart. So also in this module, this includes an amino acid chart or a codon chart. You will need this, and those also have amino acids listed. So you have an amino acid attachment site, and you also have an anti-codon site. Now remember how we called our non-template strand the coding strand. Well, that is also semi-related to our anti-codon, because remember, your coding strand or your non-template strand is exactly what your mRNA looks like, except your mRNA has U's instead of T's. So RNA can often take on this like 3D structure, right? So normally RNA is single-stranded, so it only has one strand. However, if you look at this particular diagram, RNA can actually take on kind of a 3D look through base pairing. And this is crucial to how tRNA and mRNA function. Okay, so this is just another look, probably an easier look on the left here about what a tRNA or a transfer RNA looks like. You've got these different regions. You've got this red region, which is highlighted here, a green region. And then as you can see, these base pairs we're pairing together. So that's kind of how it gets this like 3D look. You've got an attachment site for that amino acid, and then you've got your anti-codon loop. Now, this anti-codon portion is responsible for recognizing specific sequences in your mRNA. And your anti-codons are going to be complementary and anti-parallel to your mRNA codons. Now, what do we mean by codon? codons are this kind of 3 base pair site. So the way that transfer RNA reads things, or the way that mRNA is transcribed, is in nucleotides, like a set of 3. So AUG here, for example, is 1 codon. The next 3 will be another codon. So you can see that this transfer RNA is coming in to match up with the specific codon on the mRNA. And once they do that, you can see that they bind, right? Because remember, A is going to pair with U in this case, because this is RNA. A is pairing with U here, and then G pairs with C. So it's recognized this codon site, it's paired, and you can see that it's got an amino acid attached to the end, which is known as methionine. And we'll talk about how you get more than one paired up. So if we're dividing translation into 3 stages, just like we did transcription, it has also 3 stages that sound very similar, right? Initiation, elongation, and termination. So your start signal, remember we talked about these regions of genes having a promoter or a start region. That is basically a region that codes for a start codon. So your start codon in mRNA is going to be AUG, and you need this to assemble the ribosomal subunits plus the mRNA, and then initiation factors kind of help get everything started. Then you have elongation. So this is a 3-step cycle that adds 1 amino acid to the polypeptide. So if you remember, a polypeptide is many amino acids put together, and that forms a certain protein. And again, you have, just like you have initiation factors that help kind of start things along, you also have elongation factors, which kind of help keep things going. You also have a stop signal. So this will be your stop codon or your terminator region, right, in the mRNA. And we have 3 different stop codons, and we'll talk about those particular codons. And just like you have elongation factors to keep the process going, you also have release factors to end the process. So that's basically kind of our 3 stages of translation. And it's all about, remember, getting, from your DNA template to an mRNA copy and then translating that into a protein. Now, recall from earlier in the class, our amino acids are our subunits of proteins. So that's what we're building here. We're building a chain of amino acids. So first things first, you have your small ribosomal subunit. Remember, your ribosome is composed of 2 different parts. You have a big, large subunit, and then you have also a small subunit. And this is going to bind, the small subunit will bind to your mRNA and initiator tRNA. So you can see here that our mRNA is now bound to our small ribosomal subunit. So these purple things here are initiation factors. They're just helping everything get started. And then you also need the anticodon AUG. So you can see down the line here that we've got an initiator tRNA anticodon AUG. All right, it's carrying the amino acid methionine, also abbreviated as MET. So again, same thing here, right? Methionine has now bound itself to AUG, right? So UAC binds up with AUG. And now your tRNA, your transfer RNA with methionine is now in what is known as the P site. And we'll talk about these different sites in a little bit. And it's ready to begin adding those amino acids. So you'll notice that you've got another transfer RNA coming in with another amino acid that is trying to match up to our next codon region. Remember, these codon regions have 3 different base pairs, right? They look at it in 3s. So you can see here that valine is actually going to line up nicely, right? So C pairs with G, A pairs with U, U pairs with A. All right, so you can see valine is lining up for its amino, the amino acyl site. All right, so we were talking about these different names of sites. Well, during elongation, in this large subunit, you have 3 different sites. You have the amino acyl site, You have the peptidyl site. So peptide, you can think of now things are bound. You've got an actual amino acid that's getting added, and then you have the exit site. So after things are bound, the transfer RNA will leave, and it's leaving at the exit site. So your P site, it's going to hold the growing polypeptide chain. So you guys just need to know it as the P site. The A site holds the tRNA. It's kind of essentially waiting, right, to transfer it over to the P site. So it's carrying the next amino acid to be added. And then the E site is your exit site, and that's where it kind of gets rid of tRNAs. You can kind of think of this as like a, you know, like a chain, what's it called? I don't remember. I don't remember what I want to say, but this is where the E site is where your tRNAs are discharged, and they leave the ribosome, presumably to go get, you know, a new amino acid attached to it. So first things first, codon recognition. So that's where your tRNA complementary to the next codon, right? So CAU is complementary to GUA. It enters the A site. The anticodon of the tRNA then base pairs to the mRNA codon. So you see this will enter the site, it will pair up. Then you have peptide bond formation. So basically this pairs up next to it, and you can see now that that methionine that was on our original start codon here forms a peptide bond with valine, which is our new amino acid. All right, and so then once this bond is formed, that original transfer RNA kind of moves out of the way in the exit site, and valine will slide over and take its place. And this continues to happen. As you can see, there's a new one coming in, which is arginine, and it's ready to do the same thing. And this is going to continuously happen, and elongation factors are going to help move it along as well. Okay. Now, this is where you get to actually translate a specific mRNA sequence into an amino acid. And I will have a different video on how to use a codon chart and show you guys how to do that. So for now, just know that this is coming back, and I'll show you how to do that. So remember, our mRNA is read in 3 different reading frames. So this is like one codon is 3 different nucleotides. So if you were a ribosome trying to read this mRNA sequence, it would look something like this, where you have 1 fat cat ate 1 fat rat. It would not read it like this. And you can see how if you just shift it, right, just 1 over, then it doesn't quite make sense anymore. Same thing here. If you shift it again, still doesn't make sense. And that's what those start and stop codons are essentially for, is to help the ribosome read it in the proper direction. So remember, our start codon or our promoter region, right, tells us where to start reading. So in this case, AUG is going to basically be kind of that start codon. It's going to tell us, all right, you need to start reading stuff here. So it's going to set the reading frame. And that's how you have the proper amino acid gets added, because base pairing between the mRNA codon and the tRNA. So if you didn't have this, you might have a reading frame that looks more like this, right? And that doesn't produce something that's usable. I also want you guys to know that an mRNA can be translated by multiple ribosomes at the same time. And so when this happens, you end up with this kind of chain, right, with multiple ribosomes on it, and it's known as a polyribosome. Okay, and so just because you've translated a bunch of amino acids, you form some polypeptide chains, that doesn't mean you get the proper protein. So you have to do some post-translational, so after translation modifications. So some of these will get sent to specific locations in the cell. You might add some sugars onto the end, or you might add a phosphate. And when you add a phosphate, we call this phosphorylation. If you add a sugar, we call it glycosylation. You might chop up the protein into little bits, which is proteolysis or proteolysis. And so when you do this, this kind of changes what the protein looks like. And it might change its function in the cell. That's all you have to really know. Okay. And I also want you to know that changes in single nucleotides, right? We talked about specifically cilantro and how a change in a single nucleotide kind of changed whether individuals had a preference for this. It can also have really big impacts aside from like whether or not you like cilantro. And so, for example, a change in a single nucleotide determines whether you have a normal red blood cell versus a sickle cell hemoglobin. So you guys have probably heard of sickle cell anemia. There's a lot of problems associated with having that disease. So your red blood cells don't carry as much oxygen. And there could be issues and sometimes even death associated with this condition. And so basically what's happened here is you can see that there is a single nucleotide change. So instead of having an A here, it has a T. And so this is going to actually change what protein or what amino acid gets added in this peptide chain. So instead of GLU, now you have a valine here. And so this is going to change the actual gene. So where now instead of producing a normal red blood cell, you're going to produce a sickle cell. So one change can have big, big impacts on an individual. Now, not all mutations are going to have the same effect. So this mutation happens to really impact what these cells look like, but other mutations, they might not actually have a big impact. Okay, another example of a mutation that has a big impact can be for cystic fibrosis. So in cystic fibrosis, they've actually deleted just 3 nucleotides in a protein coding gene. So instead of it being this long, right, we're missing 3 nucleotides. And as a result, our peptide chain looks like this. So we're missing this middle amino acid. You're missing PHE. And as a result, the CFTR protein, which is a transporter that helps, pump chloride ions out of the cell, is degraded. So it doesn't actually do its job, and it actually results in the condition known as cystic fibrosis, which is a respiratory condition that often you need a lung transplant later in life because it really disrupts your breathing. Okay, so where are we now at the end of translation? So remember, translation is all about building polypeptides from mRNA templates. As opposed to transcription and DNA replication, our templates were DNA in that case, but in translation, our template is RNA. Translation is going to occur in the ribosome, right? And we're building polypeptides, which if you put enough polypeptides together, that makes a protein. Our building blocks, again, are amino acids, right? Because we're all focused on proteins. And then in this case, we're reading our template 5' to 3', but we're starting with what is known as the N part of the chain to the C part of the chain. All right, and then our template, of course, is messenger RNA. So that's what we started with. All right, and then also please watch the video on how to actually translate your mRNA into amino acids.